Okay, welcome everyone. Thanks for taking time out of your evening to visit with us tonight. I'm Brad Unsicker. I'm with Win Medical and Apex Medical. I started Apex Biologics about 10 years ago, and we started out with autologous products and slowly started wanting to become a full-service regenerative medicine company. We started adding allograft products to our offering. And we ran through a whole bunch of labs over the years, and we continued to look and find, tried to find a lab that we could trust, a lab that did everything right. And just in the last couple of years, I reconnected with a good friend of mine named Vince Liguori, who will be our speaker tonight. And Vince is been around in the regenerative medicine space for a long time. He was instrumental in the operation of the largest regenerative and anti-aging meeting, and he managed those, made sure they ran properly, and did an amazing job with that. But Vince is now the current CEO of Ageless Biotech, and one of the co-founders of L2 Bio and Cresca. Ageless Biotech is a company committed to the advancing of adipose-derived stem cells and therapies, and currently has ongoing FDA-approved phase 2B-3 INDs, and it's involving a multi-site 380-participant clinic targeting osteoarthritis utilizing adipose-derived stem cells. And Cresca is an off-the-shelf, ready for immediate use, live adipose-derived cellular allograft that is FDA compliant for subcutaneous injections anywhere skin and fat naturally exist. Cresca is weeks away, he's announcing this, weeks away from starting a clinical trial for the treatment of Crohn's disease. He also recently has brought an EV product to market, which we'll discuss tonight. So the way we'd like to roll tonight, we're going to turn the time over to Vince, and he will hopefully answer all your questions before we get to the question and answer portion of our segment. But we're going to turn the time over to him, and he's going to educate us on these adipose-derived products that he's brought to market, and the reason why they are so good, and why they are ethical, and why we are able to use them. And he will lay this out, and then we'll open it up for questions. If you want, if you could type in your questions, we'll start lining them up toward the end so that we can hit them and not waste any of your time. So with that, we'll turn the time over to you, Vince.
All right, great. So I really appreciate the introduction. I'm gonna turn off my face so nobody's scared, and I'm gonna go right into the presentation. And this was the order I was asked to go over. So don't get scared when you see the compliance stuff pop up right away. But you're gonna see the compliance stuff pop up right away. If I can find it. All right. So let's just pick a field here. Yeah, guys, I'm trying to share the screen. It always works perfect when we're doing the thing, right? Share screen, there we go. All right, guys, so I'm going to open up this PowerPoint. We're going to go right at it. I hope everybody can see that. Hide my little thing here. And I'm going to go to the full screen. There we go. So I'm opening this up in the FDA regulations. So I was asked to, this is kind of the order I was asked to do it. I don't always start off this way, but I'm going to go right into this because this is always an issue and people are concerned about compliance. And some people are just not as concerned. But the way things are going in the market, you should have more and more concern about it. Now, what we believe is we have a more powerful, better, well-studied product, and it is compliant for certain uses. And I'm going to go over why right now. And I want to keep this agnostic to products right now at first. So assume there is no product being promoted here. And a lot of what I do actually is on the clinical trial side too. So it takes up a lot of my time on the science side. These byproducts are things I helped invent, but I'm not involved in the day-to-day operations. So just everybody understands that. Okay, so when you say I'm a 361 or 351, and I'm assuming most people know what that is. 361 is kind of an exempt product that falls in a category where the FDA does not have to. You can actually market it for certain purposes. It's considered homologous in use, and it doesn't exceed what the FDA considers minor manipulation. It's supposed to act the way it does in the person, the way it does in nature, and it's supposed to be from the same source. So bone from bone, cartilage from cartilage, fat from fat, skin from skin, cartilage, or etc. So I think we all understand that, and we're going to make it very clear here. Products that are altered in any way are grown in a lab in certain ways, particularly in a bioreactor scenario where you force massive growth, or synthesized in any way, or mixed that in combinations other than a few acceptable sources like saline and cryopreservation substances are more than minor manipulated according to the FDA. Now remember, the FDA may have opinions that some of us disagree with, and we may think that their opinion is narrow, but in some circumstances right now, I'm going to tell you that their opinion is more liberal than mine and maybe most of yours, and this will be a little amazing when I get to that part. Okay, so if you look at this first sheet here, the FDA, right, requires the FDA's opinion, we'll say, right, and that's whose opinion matters. This is what the FDA publishes as a source of acceptable homologous sources. So what that means, if I look at the very first one, it says number 1, CBRE, and you go down to where the little bullet points start: bone, ligament, tendons, fascia, ocular tissues, skin. By the way, Cresca is classified off to the right here. You'll see a subcutaneous skin fat, and we don't market it for systemic use. And we'll get into all that in a second. I'm going to read that after I go through this list. Vascular grafts, amniotic membrane. I'm going to drop to the end. Cord blood. I'm going to skip that next one because people are going to make fun of me that are on this call, and I'm going to skip. I guess I'm going to skip the rest, and I don't like to pronounce the last one. But what I'm trying to say here and point out to people is fascia can be harvested from one person. Certain standards have to be followed, minor manipulation, etc., and reimplanted into another person. That would be considered homologous use. Tendons, I could take tendons even from a cadaver as long as I follow certain, and most of it does come from cadavers, like cartilage. In almost every single nose job, there's cartilage, and that donor is a cadaver. That is considered homologous use. That does not have to be FDA approved. It's actually, if people don't think of this, but if you ever had rhinoplasty, you're likely to have some deceased person's cartilage in your nose. Sounds a little creepy, but it's true. So skin is also classified as fat, and fat in many ways is classified as skin. So I'll read this real quick to you. Cresca is classified as white subcutaneous skin fat. Is not marketed for systemic use and is a live adipose-derived cellular allograft marketed for intradermal, subcutaneous, and topical use where skin and fat naturally exist. Adiposomes are byproducts of the same adipose allograft source. So very few companies will come and say, or will say at all, that I'm marketing a product for subcutaneous, even intradermal use. Subcutaneous use is not topical. It's not cosmetic. It's not Avon or any type of makeup being sold as exosomes. It is an injection. Right, and we'll go into that into the next screen. So here's what the FDA published, and if you're not familiar with the guidance, it's 21 CFR 1271. It's right up here, but that's really the guidance that was put out in 17, and then had a deadline of December 2019, and they moved the deadline again. It ended in I believe it was May 31st of 2021. And this was that period of time where we had a little bit of leeway until we had to comply with some of these guidelines. And what I did here is I highlighted what's important to this conversation. Okay, minimally manipulated is what everybody in the world is going to tell you, right? And it's probably true, I think, in most cases. Most people out there making biologics are following that guideline. I would hope so. 2, the HCTB is intended for homologous use only as reflected by the labeling, advertising, and other indications in the manufacturer's objective intent. Okay, here's where almost every product gets it, at least according to the FDA. Now, we can all have differing opinions. I have some differing opinions here too. I'm also not the FDA. Now, objective intent. You cannot have a homologous product without being able to demonstrate a homologous use. So that means fat to fat, skin to skin, bone to bone, just like that list. It's that simple. Now you can make arguments for other products. I do believe there's some products out there that have a homologous use and intent. And then if you do not believe this, you can go, or you want to argue with the FDA, or you have a different opinion, you think you're right, or you want to ask a question. They do have a mechanism for that. We can go over that at a later point, or you can ask the question. It's called a TRG. And then the more advanced, which is virtually impossible to get, is called a RFD. That stands for Request for Determination. Then there's the drug route, the IND, and a device route. And that's not the point of this meeting. We can have another one if people are interested, because I do know all about that, but that would be another topic. So once again, homologous use reflected by labeling, advertising, other indications, and manufacturer's objective intent. What are you going to do with it? So if somebody says my product is homologous, my product is compliant, give me a compliant use, right? With Cresca, we can definitely do that. We can make lots of arguments for the Adiposome product too. But I'm just telling you that Cresca, we can do it. And we can go over some more of that later too. Then this other section, either. So an HCP does not have systemic effect and is not dependent on the metabolic activity of living cells for its primary function. Okay, we can comment on that in a second before I go to the or. So that doesn't mean you can have a live cell product. And I'm going to talk later, or I can talk now maybe briefly, why actually having live cells is more of a compliant product, even though I do believe there's great ambient products out there that are compliant as well, because the live cells act as they do in nature, but you cannot market that these live cells are going to cause, your heart to completely repair in the future, because we're depending on differentiation and cell replication. So replication and differentiated what stem cells are supposed to do, right? Or MSCs or certain cells or multipotent cells. You cannot say, I believe this is going to do this 3 weeks from now or 2 days from now or weeks, because you don't know. And it's those claims that get you in trouble. Because if we go back to the top here, it says labeling and advertising for indications. So if I'm saying that my stem cells and my exosomes are going to cure your ABC disease, and it takes time, and it takes a couple of weeks, or waiting for them to act, and then putting it in IV, that is not compliant. I think most of us know that on the call, but that's what they're concerned about with the marketing and advertising. And that's not off-label, because it's not on-label, because you don't have a compliant use. So you do have to be careful. I hope I've kind of illustrated that. And then when I go to the or, which is number 2. So the first one was can't market systemic effect, which is what I just gave you an example of, or metabolic activity of living cells. We don't know what's going to happen in the future, right? The metabolic activity, we can't predict what's going to happen in the cell. We can't use this as the basis of the marketing for its primary function. So we know that it has that. We know that it has proteins, and we know that it has elastins, and we know that it works like this in nature, and we can put it in the same area, and we can hope it. But you can't make the claim unless you have absolute proof, and it's either approved or you have some sort of designation like an RFD from the FDA. Okay. Number 2, the HTP has a systemic effect. Now here you can have a systemic effect, or it's dependent on the metabolic activity of living cells for primary function, and is autologous use, BRP. I don't have to, I think most of you know what that is. It is for allogeneic use for first-degree blood relatives. Now that makes sense. Now here's where I think most people don't understand this, because I have a lot of people that tell me that this doesn't exist. But it's true, because here it is on page 3 of the 1271 CFR guidance under section 2, number 4B. Second-degree blood relatives. So for autologous use, now we're not talking about autologous products today, but this is an example where the FDA can be more aggressive than what people think. They consider that for all practical purposes, not autologous, but compliant for allogeneic use. Next thing that people wanted me to go over is donors. And I believe that at least with the Cresca product, the Adiposome product is actually exactly the same. We probably have the most intensive donor screening process. We do screen donors. We do take people. We have to take people over 18. We're looking at cellular age. We're looking at, a bunch of different factors. We're testing for everything you see on this list. But more importantly is we've added actual tests of tests that were already being conducted to test for, and let's give you an example, a nucleic acid test for HIV, to test for the probability of people contracting HIV 3 months from now or whatever it is. And there's a score scale, and we throw those out. The other thing we've done with Cresca is the second, it's actually more than 2.0, but I call it 2.0 right now because I had another experimental product before. But it's the 2.0 of another product we had. And after we had the FDA audit, the FDA suggested, because they know we're on track with a homologous product, the way we market it and the way we make it. However, they wanted us to add a couple more tests. So we did they suggested we do a recall and add these nucleic acid tests. And they also wanted us to use, and we were using LabCorp. I like LabCorp too. And, people have different opinions about LabCorp and Crest and stuff, but it's very accessible for people. They wanted to use a donor specific laboratory that just tests donors that the FDA itself uses or recommends. So we said, okay. At first it was way more expensive, and now they've become reasonable like LabCorp because we send so much volume to them. Numbers detect everything. So now they've been very reasonable. So at first it was very expensive. So we did a voluntary recall of that product. So we actually did a recall. We took that product back. We paid for it. We destroyed it, and we created the new product, hence Cresca. The FDA then terminated the recall. And guess what they gave us? I only thought they were a legend in biologics, especially people that make cellular products in the biologics and the 361 space. They gave us a perfect audit. So now we actually have a perfect audit and a terminated recall with the FDA. And we're working on a bunch of other things, which I can't talk about right now, but it's very, very exciting. So this is very important. And then with the adiposome donors, so with adiposomes is very interesting because now we have an autologous growing project and a banking project. And what's happening is we have unlimited donors. So we cherry pick the ones we like. And, we offer them incentive, and they go through the whole donor testing process. And we're only looking for people between 18 and 40 years old. And we're looking for a certain body mass, and we're looking for non-smokers. And we're testing them to make sure, they don't have cooties from kissing that girl in the 4th grade and anything you can think of. And the standards are very high. And unlike a lot of the perinatal products where it's just a questionnaire, and you're still supposed to do all this stuff, but we are not growing from the same placenta 4,000 times or the same, organ or whatever, any kind of weird science you can think of. The adiposomes are produced from 3 specific donors right now. We're adding 2 more, and we've all tested and evaluated, and we'll get into that too, which is pretty cool. So we know we've got a really consistent product, and I'll get in that in a second. And actually what we're doing is we're not diluting them. We're taking that straight from this protein-based media culture that uses virtually nothing that's, you would consider it's kind of a natural stuff. I'd like to talk to people about it. I actually sent the formula to a couple of people, and my partners almost murdered me because I think even with the formula, this is actually not that easy to do, because I started at this, at least for the exosomes, in 2018, and I always wanted to have adipose-derived exosomes like an off-the-shelf adipose-derived allograft, with this huge, not just MSCs, with the entire cellular family. So I'm going to move on from this. This is just an example. By the way, just like this previous slide back here, I have this document from the FDA. It's printed. It's from the FDA. Whoever invited you to this presentation can send that to you. I have this, and I have the entire guidance. If you don't have it, it's pretty easy to find. Here's a link. You can find this. This is actually on page 3. So you go to page 3, and it's part of section 2, which starts on page 2. Ironically, right? So with all that said, here's and this is kind of a Cresca plug, but this is the single most important thing when you're injecting cells after implant. I'm not talking about metabolic activity. I'm talking about survival. Okay. So let's just talk about survival rate. For things to work, they need to survive. For things to work over time, and especially stem cells, they need to be live. We're not going to promise that live cells are going to, cure anything. We're just going to say that the proteins and whatnot are going to work longer. And that's the real mechanism, is keeping these things alive. I do believe they're going to differentiate into new cell types and replicate the correct cell types. We do know it's factual that the telomere lengths in adipose-derived stem cells are up to 3 times longer than other stem cell type sources. We do know they live much longer, and we do know that in reality, they're much younger because of the lifespan of when they're harvested versus the lifespan at the end of a baby's life, because you're harvesting things at the end of the lifespan. But they survive longer because they also can handle oxidative stress and a lot of other things. And that's why I want to put this article up here. I'm not going to read the whole article. We're going to also supply this to anybody who wants a copy of this. And let me just read the statement here. So after implant, this is the title too. After implant, adipose-derived stem cells have vastly superior survival rate than perinatal sourced stem cells. I think most of you know what perinatal sourced stem cells are. It's pre-birth, cord, amniotic fluid, etc. Amnion. So that survival rate's everything. Now, not only am I saying that, I'm going to start here with the references. There are 28 references here. These are peer-reviewed sources, articles, studies, clinical trials. You can check every single link. And there's actually even citing inside of the article itself. It takes a little while to do these articles and to make them 3-4 paragraphs as opposed to 400 paragraphs or 40,000 words, because we're trying to be concise. And by the way, it's nice that you can read these sources, and we can provide that on a bunch of different topics for scientific purposes mainly. But just so you know, all right, I'm going to move on now. And what my point is to get through this demonstration is, or this presentation demonstration, same thing, right? Is, expediently as possible. I tend to go fast, so I slow myself down sometimes, repeat things, just so people understand. And this gives us more time for questions and answers. Okay. So now exosomes. So this is probably the most common question. How many exosomes are in there? How much do I get? How do we know it's accurate? And I'm going to go over measurement types. All right. I don't know anybody that uses all. Actually, I do know somebody that helped me through this. He runs a pretty good sized lab. He's got an Italian last name, and it rhymes with Edie, because I need help from other people too, right? And I do have a FDA consultant, and they have a whole team. So, and I did call Rooster Biotech. These are all really smart people. So there's people that make all these other products, including some, and I believe in umbilical products too. This is just a separate product. What I'm trying to tell you here is that these measurements, someone or somebody employs them for different reasons. And we're going to go through them so that you understand, and then you can ask more intelligent questions. Once again, I don't expect anybody to memorize this. I forget the words all the time. I've got it in writing like everybody else, and most scientists too, probably. And but we'll supply this to you too, so you understand. So these are the most common measurements, and it's really 6, not 5. I forgot the most coveted, at least by the FDA. They say they're agnostic device, which is number 6, and we'll go over that last. All right, and I don't have as good an explanation on that, but so here we go, and this is just the coolest name: atomic force microscopy. All right, Buck Rogers, I guess, right? So we're not going to read all this. This is going to be very high level. So this is really valuable for distinguishing varying sizes and morphologies of exosomes. Now, there's a lot of reasons why you want to do that. And once again, we could just highlight them real quick. But, you're looking at the sizes, and you're looking at, what they turn into, and you're looking at applications in the future. All right. So this is good for measuring sizes of exosomes. And it's also the, how would you say, consistency. So all positive stuff. And that was probably a bad explanation, but we'll whoever invited you can make all this available to, including the presentation, but you'll want the articles. So this is called NTA. You may or may not hear of it. A lot of places that have exosomes don't like this test. I'm going to tell you why. Because the NTA test gives you a very accurate measurement, or probably one of the more accurate measurement of the amount of actual exosomes you have with intact lipids. And you can get farther and go after, lipid bilayer membranes. So, but this system discounts EVs or extracellular vesicles that don't have intact membranes or lipids. So what happens is they're really not exosomes at that point, or even EVs. So those get thrown out, so they don't go into the count. What it does is it uses like little reflective lasers, goes up and down, and it's pretty cool. If you ever watch a little demo of it. I don't have one. I wish I did. I did have one, but it keeps destroying itself because I think it's proprietary when they show it to me. So I've tried to copy it; it won't let me. And but then what happens is then it uses really complex math algorithm, and it does sample sizes, and then pulls it all together and gives you a very accurate account of what you have as far as exosome counts or EV counts in a certain range. And it can go accurately from 30 to 50 nanometers up to 300 nanometers. The problem when you start going over 200 nanometers, I mean, you have them and they're probably just as good. I heard another competitor actually lecture very good about the size of the exosome, and maybe the 275 nanometer exosome is the best one because it's got more proteins, and it makes sense to me. But the reality is they're like little bubbles, and they eventually burst and just become scattered proteins with probably non-functioning mRNA. So, but let's just go over this. So that this NTA is contributes to the accurate quantification and characterization of these extracellular vesicles. So what it's really strong for is size and concentration. And what happens is a lot of people don't use this measurement because they don't like the outcome. Because I say I have 1 billion exosomes or 5 billion, and NTA comes through and says you have 6% of that. Now, on the clinical side, we've tested a lot of companies, and I can't name them, and we're working with some of them right now. And some of them I never heard of, and some of them you have, right? We all have. And we typically, when we run it through NTA, get 6 to 8% of what they're saying they have in that vial. So it doesn't mean we have a bad product. It means we're taking 10 of their vials to make the same amount that they claim they have, at least the way it's going to be looked at for a trial. The next one is a microfluid analysis. Okay, so once again, I'm going to only do high level, highlighted the high level things. And each one of these, to get it down to a paragraph, was very difficult because I have 5 to 6, 7 page definitions on some of these. All right, so this just allows for isolation of exosomes and the detection of molecular biomarkers. And we're going to go over common biomarkers, but that's very important because what you want to do, now it's not giving you the great size range in intact lipid, and I'll tell you why I like that NTA probably as good as some of the other ones. But this is a really neat, once again, analysis, and it's just something we're saying that's an exosome, right? And we'll keep going here. Number 4, immunomodulation detection. Okay, valuable insights into molecular characteristics. Okay, great. We want to know those characteristics. We want to put those studies out. Once again, probably not something that's going to help any of us. We can't say, or we can't stress the media and cause, at least legally we can't do this, and cost effectively nobody's going to probably do this. But that's really for a drug. Now, some of these great places that even have INDs and are making exosome products that are being used topically, and however it is compliant or whatever the argument is, I'm not going to get into that with the exosome, but what I'm going to tell you is this is really cool science, but it's probably not anything. It's cool for us to know that our exosomes probably do this, right? Because we're, using them. But the reality is this is a measurement that would not be something that I would be that interested in right now, although I'm interested in it. Not if I'm buying the product. I'm looking at the volume too, just like everybody else. And this is probably as common as everything, and it's really one company dominates. It's called the Jess, the protein Western Jess. So it's really Western blot analysis, and this does something that I think is necessary. It does a protein analysis, and then you're trying to look at the biological function after the protein analysis, and it gives you lots of cool things. And this is cool to know. You may want to know that's how many exosomes how many proteins are in there, what's the concentration, and then you know that these proteins in combination is good for that therapy, etc. And that's really neat science to put out there. And I think that's very valuable. Number 6, and this is the one I was going to leave out, and I said I'll put it in at the last second. This is the I-zone. The I-zone is probably, it's kind of like the standard of what the FDA and other people like to see. It's not as expensive a machine as you think, but they're actually probably on back order. They were on back order when we tried to buy one. The I-zone, it basically uses NTA and this crazy name, which I always thought was cool, tunable resistive pulse sensing. It detects the changes in electrical resistance as particles pass through the pore. Now, I didn't get into all of this, but this kind of combines 2 technologies, and you get the most accurate measurement of volume and size. Right now, of course, you're going to go, and there's a really cool microscope out there called the ONI, and a couple companies have it. The ONI actually combines the, I'll go back here. The ONI actually, and you don't have to use them all. And I had a really good demonstration by a person 2 Saturdays ago, and I've seen him before, but he showed me how he could check, whether the bioreactor that he's using is creating good stem cells or causing mutations by showing them clumped together. And it was a really, 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 really great demonstration. And I would challenge anybody buying exosomes from anybody, ask their scientist or ask a rep. I mean, how do you mitigate, for mutations and alterations, especially with TP53, which we can go over in a second if you don't know what that is, or you can ask whoever got you here, or TSG 101, which we are going to go over, alterations and mutations in the cells. What's your policy? Because a bioreactor can be, and it may be, the gift of God to help us all heal in the future, because you can produce mass amounts of these cool little miracles. But that lab has to have how does that lab protect against those problems it causes too? Because, the star that burns brightest, is also the hottest, right? So, and that different than the real analogy, but I like that one better because I just made it up. So I'm going to claim it. But the ONI it's called it's not micro, it's the ONI Nano Imager, ONI Nano Imager, which is kind of a cool word. It probably doesn't spell it. So atomic force microscopy, it has NTA nano tracking analysis. You don't have to use all these. So some of the people that only don't use the NTA part too, or they have a separate NTA analysis because they don't always like what the NTA says, because it might say you don't have a lot of exosomes, right? It might say you have a ton. Most of the time it says you don't have a lot. Microfluid analysis, immunodetection, and the ONI has all 4 of those, which is pretty cool. The ONI is also around $300,000. We just went over protein plot analysis, and I'm not going to go over the individual types of staining and stuff. So another way, they do all this is they stain things in different colors, and it picks up different proteins and different markers. Because if you want, I don't want to sit on this call for 3 hours. This is high level, and you're going to copy this. And if you want, you can Google it, and you can Google any of these things. You're going to get 10, 20 pages on each one. And if you read 10, 20 pages on them and you study it 4 or 5 times, you're going to know more than me anyway. So then that's the big question, right? This is how we get to the bottom of it. So this is what people want to know. How are metasomes measured? All right, and how many atosomes are in a vial? So I'm going to tell you, we've played with all of these, and we use it for research purposes too. And we're going to add more. And we use a couple of those labs we talked about. And thanks to Rooster Biotech as well, and one of the other labs I talked about, we're doing more research now. That's not benefiting us here right now, and not for purposes of this discussion, but it is really cool. And it's good to know that the people we work with are doing this kind of stuff. But how are they measured? So we do a protein analysis, and we also have a giant heat map we're working on, number 5 that we're working on. But what we really use is NTA, and we look at TSG 101. And the reason why that is, in TSG 101, we use 2 or 3 of those machines. I was told if I tell people which machines, even though I just took this up, but TSG 101 can be measured multiple ways. And I'm going to explain what that is in a second here. And we're going to go over a lot of this stuff here. But atosomes are measured using NTA, and we do a TSG 101 analysis. So what is TSG 101? So TSG, it stands for tumor suppression protein or tumor susceptibility protein. It's phrased both ways. I get confused, but the most common one I see all the time is tumor susceptibility protein 101. And that looks, every EV, it's essential in the formation. So how you have an EV if it has TSG 101, and we use the NTA. And how many atosomes are in a vial? Well, I'll tell you. We do not use saline. We do not. And by the way, we're not advanced enough. We're not advanced enough. We don't have the labor to do it, even though you can cut the cost way down. It's just for us at this point in our in the successful manufacturing of or help manufacturing of the Adipose product for we don't use saline to volumize. So we're not creating salinosomes, which saline serves its point. We're actually taking it from incubated media, and we're leaving the media in it. We're not using a bioreactor either. We're growing these naturally, and we are we are adding multiple donors for a variety of reasons, and we're trying to keep it kind of the same consistency as the Cresco product. We're using kind of the same technology. And by doing that, the counts were insane. I was shooting for, and I was talking to Brad about this, and it was probably, I don't know, months ago now, and I was shooting for 50 to 100 million exosomes per 2 ml. That, I mean, exosomes that I can validate with the NTA. I just went over NTA because that's not so easy to do, right? First of all, our PhD called me. I thought she was, I know she doesn't smoke pot, but at that point I thought she was a pothead and doing mushrooms or something, right? And she gave me a number, and I didn't want to call her. So we checked it 3 times. We got on the phone with Rooster Bio. I got on the phone with another lab. We all looked at the measurements. We did it again. We did it on 3 samples. We're adding 2 more, and not just lots. We did it on 8 lots. We did it on 3 different donors in 8 lots, and we're adding 2 more because eventually now we've got a real, I think, research gold mine. And maybe it's 2 years from now, but we're going to start stockpiling these things, right? So per ml, 800 billion. We can supply you that proof too. 800 billion per ml. He said these currently are sold in 2 mLs. We are making 1 ml. So that's 1.6 trillion adiposomes. We're calling them adiposomes. It's a great name. I know. Adiposomes per cc. That's a lot. And we also know the survivability rate and some of the other things and the other exosome qualities are superior in these products. Now, what I don't have in this, I'm going to go over shortly and we'll open it to questions, are common biomarkers. And I don't believe I have it in here. I thought I had it in here, but I don't think I do. What I want to do is go over some common biomarkers that they test for with exosomes too. And I'm just going to go up over them real fast. So I'm going to turn this presentation off. Or actually, yeah, I'm going to change my screen real quick. And I'm going to turn this presentation off. And I'm going to find the, I'm going to stop sharing so you can see this great screen right now. And I'm going to find my little cheat sheet and the biomarkers, because I have to memorize the biomarkers like everybody else, right? And I know a lot of them, but there's some I still have to memorize. And here, you guys give me 1 second. We're going to open it to questions. Sorry about the little delay here. I think it's just important to know these, and we'll send this to you too in writing, so you can look them up because it's kind of cool. All right, so I just went over. So I just don't want to screw these up. So TSG 101, CD63. You know what? What I should do, because people aren't going to know any of these, I'm actually going to provide this to everybody in writing, because I don't believe there's a test and anybody's going to memorize this. I think it's probably more important, Brad, if you disagree with me, let me know, to just kind of, we can supply this to anybody that's looking for it. Well, we're interested in a CD63. There's a few other ones, and TSG 101, because every exosome needs to have TSG 101. So if I finish with anything, we have a compliant product. We've got more than a compliant product with Cresco, and we have, I think we have the best product on the market. I wasn't going to get into science. We have a product that could survive for weeks after implant, especially subcutaneously, because it's going to eat and live. And I think it's a home run. And I don't know anybody that can compare this, or anybody that can compare even the way we measure them. Now, from a scientific analysis standpoint, if you're looking at the basic breakdowns and all the crazy things you can do, there are people out there better than everybody. And there's so many different measurements and so many different analyses, and mainly analyses, that it's really hard to say who's the best. But I know a few of those people that are better than most. Brad, I guess I'm done.
Let's open it up. We didn't have any questions or in the question and answer. If you'd like to ask a question, you can type one in. I think we can open up the mic. And thanks, Vince, for
welcome. I'm trying to breeze through that because there's a lot of info, and it bores the hell out of me too. The compliance stuff amazes, like, okay, so there's something that does amaze me still. I still think it's pretty, liberal for the FDA to say, second degree blood relatives are okay for, because it just doesn't sound like the FDA to me, right? Yeah. The fact that we can use live cells
too. Let's open it up. Yeah,
okay.
Let's open it up.
All right.
Anybody have a question they'd like to ask Vince? Okay, there's one. Got a raised hand.
I get the raised hand.
Dr. Colvin.
You hear me?
You can hear.
You can hear?
Yep.
Yeah,
Perfect.
so the adisome is mesenchymal stem cell, or is it exosomes derived from the stem cell?
So exosomes can't be derived from a stem cell. Stem cells produce extracellular vesicles, which 90% are exosomes, they're communication vesicles, basically. So in order to grow them correctly, they're from mesenchymal stem cells or medicinal signaling cells, whichever side of the camp you're on. They're adipose-derived MSCs.
Okay, so they are exosomes derived from the adipose-derived MSC.
Correct.
Okay.
All exosomes come from stem cells. Or another cell type.
Yes, they are cell-to-cell communication.
Correct. So what we could do, because I know it's a little lengthy, we can give this presentation to people and we can send it out to them. I mean, all this stuff is okay for distribution.
All right.
And I went through this with our FDA consultant and attorney, and we're not saying anything about, well, actually, half the things we're saying come straight from the FDA, probably more than half.
Go. Vince, we appreciate your time and all your hard work. It's you always amaze me with your background and knowledge. And there also is, if you're interested in, we do have in trying and using these exosomes or MSCs in your practice, we do have some introductory pricing that I think will be to your liking, and you will be able to utilize these 1.6 trillion EVs into your patients, and you can see the amazing results that'll come from it. But we appreciate you taking the time to hear about these products and the cutting edge opportunity that they provide you.